Project Catalyst presents Bench to Bedside Chats, direct from Building 22, White Oak Campus, FDA. Brought to you by the Oncology Center of Excellence, under the direction of Dr. Richard Pazder. Beyond these doors, the Oncology Center of Excellence, in collaboration with the Centers for Drug Evaluation, Biologics Evaluation, and Devices and Radiological Health, including scientists, regulatory reviewers, and leaders, have developed multiple programs focused on improving the treatment and cure of cancer. Project Catalyst is one of those efforts intended to help the academic scientist, university life science accelerator, and oncology startup company navigate the regulatory drug development enterprise and translate your insights into drugs to treat and cure cancer.
Good afternoon, guys. It's a thrill to have you guys here. Project Catalyst is really excited that you guys are willing to participate in our bench to bedside chat today on the CMC guidance with regards to GMP considerations for phase 1 studies. I've known a couple of you guys for 20 plus years, and I've recently met John here. And it's Dr. Metcalf, it's Dr. Weinberg, and Dr. Steffens, but I'm going to use first names here, John, Wendy, and Olin. And I just want to kind of set a stage for what these bench to bedside chats are all about. So our audience and you guys, I know I've said this before to you, but just so we kind of refresh our memories. These are, they're sponsored by the Oncology Center of Excellence, and the idea is, the audience here is towards small companies, academic accelerators, academicians who are trying to develop and create discovery and develop a drug for anti-cancer applications. And many times this particular audience has really limited resources. They may have just formed an LLC to get SBIR funding, maybe a couple postdocs and a professor who have formed this company. So they don't have a lot of experience, and many times the drug development enterprise is a complicated endeavor. And also many times the novel ideas, the creativity exists in this academic world, and these are the products that sometimes make paradigm-changing advances in anti-cancer therapy. So the whole purpose of this is to try to help encourage and facilitate those investigators to be more efficient to avoid challenges in their drug development efforts. The other part of this is to also help people realize that you guys are actually scientists. You're involved actively in meetings, in education, in developing guidance documents, so folks can see that we're really not just kind of bureaucratic box checkers, but that we're not a black box. That we're really out there to try to interact with these investigators and to help them. And then most importantly, what I really would like to emphasize is, these are informal. And the idea is to help get concepts across, to provide some granularity on the guidance documents that we'll discuss, to use those as a kind of branching point, to kind of get the oncology perspective around kind of drug development, because these are imminently life-threatening conditions. There's a little bit of a different risk-benefit assessment that goes into those decisions and those thoughts, but that we're not here to make new policy. We're not here to reinterpret regulations or make new regulations. And this is not, the idea here is not, here's the lowest threshold that you need to jump over to be able to get into a first-in-human trial. The idea here is to understand what our thinking process is, what the concepts are, how you should approach drug development in kind of an informed, thoughtful fashion, that you're less likely to end up with insurmountable challenges. And having said that, I would like to have each of you guys introduce yourself. kind of tell us a little bit about you, what you do, whether you still are in a lab, or what your specialties here are at FDA. And we'll start with Wendy.
I am in the Office of Product Quality. I've been at FDA for 24 years. I came as a principal investigator. I was recruited for my background in molecular oncology, which is specifically focused on squamous cancer pathogenesis. We had, at the time, a research reviewer model where there was a cohort of investigators, of lab-based assessors. So we ran research programs, worked in research programs, and also spent about half of our time doing regulatory review. And so I was a lab chief in that environment for 15 years, and that entailed both running my own research program, but also overseeing the regulatory work of 3 different principal investigator units, including my own. And we followed drugs from the earliest stages of drug development, pre-INDs, through marketing and post-marketing changes, all biotechnology products. And recently, we've been reorganized, and so for the last number of months I've been fully on the research side. Our research is focused on addressing issues that can come up in product quality manufacturing, characterization, and so we both focus our research on proactively addressing concerns and also responding to issues that come up. And so my particular group is looking at, focused on bioassay development to get, we'll be discussing this, I'm sure, the need for mechanistic bioassays that reflect the mechanism of action of the product. We also do consult reviews largely related to bioassays of complex generics.
Great, thanks. John?
Yes, my name is John Metcalf, and I will say that one of the neat things about this project for me is I'm meeting 2 new people, new to me, here at FDA. I've worked with Olin and known Olin for many years, but I'm meeting Wendy and Jeff for the first time during this project. So good, really good to meet you, and I'm glad to be involved in the project. I'm in my 22nd year here at FDA. I'm a microbiologist. I'm in the Office of Pharmaceutical Quality and the Sub-Office Manufacturing Assessment. OPMA is Office of Pharmaceutical Manufacturing Assessment. And my role over the years has been to perform microbiology assessments for both sterile and non-sterile drugs. And of course, the sterile drugs are those that are injected into the body or applied to the surface of the eye. And those are required to not contain any microorganisms. So my job on those review teams at FDA is to make sure that they're manufactured in a safe manner, and to make sure that they're manufactured such that when patients are given a drug that's injected or applied to the surface of the eye, that they're not going to get microbial infections from contaminated products. I mentioned the non-sterile drugs as well, and certainly those pose a lower risk to patients in terms of microbiological risk, but there is still micro risk with non-sterile drugs. And so the non-sterile drugs need to meet certain thresholds in terms of the amount of microorganisms that they contain at release, and they need to be absent of pathogenic organisms and organisms that could cause patient infections. So that's been my role over the years. Recently, in the Office of Pharmaceutical Manufacturing Assessment, as microbiologists, we've also begun to do assessments of the manufacturing facility and the manufacturing process. So it's been exciting to kind of diversify over the years and take on some of these additional review functions within OPQ. And the last thing that I'll say about myself is that I've been a subject matter expert regarding pharmacy compounding policy in CEDAR and in OPMA.
Is that extemporaneous compounding as well?
Well, pharmacy compounding, where the intent is a pharmacist is making the drug instead of a traditional manufacturer. And so we can talk about that later, Jeff,
we might avoid that subject, actually.
Well, if it comes up, I'm ready to talk about it, Jeff. But just to say that in 2010, I became a FDA liaison to the USP Expert Committee on Pharmacy Compounding, and so I've been very involved as a microbiologist in policy work that CEDAR does regarding compounding. So I think that's enough about me for now.
Olin?
Sure. My name is Olin Stevens. As with my colleagues, we're all in the Office of Pharmaceutical Quality. My office is the Quality Assessment Branch. I'm 100% a reviewer, and I've been there for 16 years in various iterations of different reorganizations. And I think John was one of the first people I worked with. I've bounced around a lot. It may have been in metabolism or neurology or psych. I've worked with about 2/3 of the different clinical divisions. But for the last 8 years, I've been working directly with the oncology and hematology review offices, and I seem to have found a home there. And we were talking earlier, I think Wendy and I have worked on a couple of antibody drug conjugate projects together, so good people to know here. So I've worked as a reviewer and as a frontline supervisor, but went back down to be a reviewer because that's where I feel like I have the most impact, and it's a good fit for my skills. As a reviewer, I've had the opportunity to work on some other guidance documents, and probably the one I had the most active role in was the drug products that contain nanomaterials, and that has been a gateway to introduce me to people in the lab, not on at Wendy's scale at the larger molecules, but small molecule side. It's been a nice complement to working in oncology, because what we see is that people don't generally go into novel dosage forms unless there's a real need. So in oncology, as Jeff mentioned, the risk-benefit ratio is a little bit different than what you see in other clinical divisions. So there are risks both for the patient, for the applicant, there's a business risk of developing a new technology that it just may not work, and it has other patient risks that go along with it. That we tend to see more of those nanotechnology products going towards those life-threatening diseases or diseases that don't have any treatment at the time.
It's interesting. I mean, nanoparticles and antibody drug conjugates have been around for a decade, but in the last few years, there's just been a explosion in the number of different sponsors who are developing various antibody drug conjugates and various nanoparticle approaches, and I'm sure that we're going to talk about that. Now, I'm going to try not to throw any curveballs during these discussions, but these guys are well aware that if I do, that they can re-guide that conversation away from those curveballs. So one of the questions that I always ask, and Wendy and Olin have participated in a number of other interactions, meetings, conferences, and I'm not going to ask these guys this question. I'm going to ask the question and I'm going to answer the question so that everyone else doesn't have to answer the question here from OPQ. But one of the questions I always ask, and remember the whole idea that these are early stage, early academic investigators, and I've been at conferences and I've been in meetings where these folks have done things that large pharma actually wasn't able to accomplish, or didn't spend as much time on it as maybe they should have. But I have seen protein degraders for fusion oncoproteins where they were not quite at drug level IC50s or binding affinities, but we're in the 1 micromolar to high nanomolar concentration, so they were getting close to what looked like it could be a real drug. I know some large pharma companies that had also tried to pursue the same fusion oncoprotein, looking for protein degraders, and weren't able to find any. So but these folks, many times will ask the question, or at least have think about the possibility: can I make this in my own lab? Right? I've done the discovery, I've created the drug, I've done the synthetic route, I've made the ADC. I found a target, the epitope. I've made an antibody. Can I make this in my own lab? And so this is a question that I would ask many times. And the answer I'm just going to answer for you guys is just essentially no. But I want to throw out, the reason this CMC, phase 1 guidance document, was developed was there are regulations with regards to manufacturing, and John will talk about that, but regulations with regards to manufacturing concepts and principles to adhere to, but I think folks were aware that there was there was a vacuum with regards to what the expectations were for CMC considerations for early stage phase 1 drug development, and that those regulations, because it is regulation statutes, were more burdensome because it's going to be a drug that's approved and utilized, in a broad patient population versus, what we would expect for early phase drug development, for a first in human trial. And I think that's that the whole idea was to be less burdensome. And I think the important concept, which I'm sure John will make, is that there aren't, unfortunately, for the academic investigator and the small drug company, there aren't small drug company regulations and large drug company regulations. There are the same regulations apply to everyone who's developing the drug. But why don't if John and all of you guys can kind of help us understand what this CMC phase 1 guidance document was, to try to make it easier, or not make it light, because that's the wrong word, but to make it more approachable.
Sure. Let me give my best shot at that, Jeff. I know you wanted to talk about the GMP phase 1 guidance document, and I'm glad to have that conversation with you. And when I read that document, and I'll say as an FDA disclaimer, because as an FDA person, I'm always thinking about disclaimers. And I did not work on this guidance, but I think that the guidance does a very good job of explaining to folks that are ready to bring drugs into phase 1 trials the GMPs, which are important for their level of drug development. And so if we compare a phase 1 drug with a drug that is coming in as a marketing application. So the company is now ready to commercialize that drug, and upon approval of the application, whether it's the NDA or an ANDA or a BLA, that drug we know is going to go into tens of thousands, many people, right? As compared to what we're talking about here, Jeff, is a phase 1 trial where we've got very small numbers of patients, right? So there's a public health risk difference there when you're talking about a drug that's going into many thousands of people at commercialization versus much fewer, 10 subjects or small numbers, right? When I read that guidance, my understanding of the guidance is that it's still very important to follow GMP, because we're talking about a drug that's going to be administered to humans. So at FDA, our first consideration at phase 1 is safety, right? I think we would all agree that drug needs to be safe. How do you make a safe drug? You start off by using good manufacturing practices, the GMPs. But the fact is, and guidance describes is that not every letter of the full GMP is appropriate at phase 1 as compared to all of the regulations that make up Part 211. And for our audience, when I refer to Part 211, that's the Code of Federal Regulations, CFR, Part 211, outlines all of the regulations that are necessary, required for manufacturing a drug. Okay, so what CDER has laid out in that guidance is that not every one of those regulations is appropriate when we're talking about a phase 1. So here CDER is, in my read, trying to encourage companies to get that clinical drug into the clinic. You alluded to this a few minutes ago, Jeff. So this is my read in terms of the guidance, and we're at FDA, and all of us know this, we're constantly walking that tightrope, right? We want Americans to have access to drugs. We also want those drugs to be safe. So we're trying to balance here in the guidance. We're trying to balance which of the regulations are important when we're talking about a phase 1 product. And the best advice I can give to our viewers, Jeff, is to go through the guidance. What I've thought about is all of our viewers have access to the full GMPs. You can go online and do a search for CFR Part 211, and you can read all of the requirements for a drug that is about to be marketed and to be marketed. And you can compare that with what the guidance lays out is important for a phase 1 drug.
It's not a long guidance.
When you do that comparison, Jeff, you'll see that the overall sections are very similar to the same in terms of sections on personnel. What are our expectations in terms of personnel training? What are our expectations in terms of facilities and equipment? And there's 8 or 9 general headers there. You can compare those. They line up with Part 211. And you can read the details that are in Part 211 and then read in the guidance what FDA is suggesting are important for phase 1. So I think that would be a good starting point for our viewers if they haven't brought a drug into...
Although pointing them towards the regulations in 211,
well,
it's pretty hard to read regulations and generate any... guidance the whole idea we write the guidance documents is to try to interpret regulations so it's a little more easy to understand, and even sometimes that can be somewhat complicated. But
my point, Jeff, is that you
see the difference
by
between
doing
the expectations.
This, you're seeing the reason that CDER published that guidance was to say, okay, we know those regulations are detailed. I'll use the word detailed. Here's what we think you should be doing for a phase 1, and it lays it out. In my opinion, pretty, it lays it out in a very understandable manner.
So I have this opinion that for first-in-human trials, for oncology, complicated drug development, is that the more shots on goal, safe shots on goal where we actually know what the target was, whether we hit the target or not, the better off we are. And I think, as you pointed out, I mean, there's some quotes here from Janet Woodcock or FDA's thoughts on this guidance document. One of the points was, the action is intended to streamline and promote the drug development process while ensuring the safety and quality of the earliest stage investigational products, those intended for use in phase 1 clinical trials. I mean, the idea was to not inhibit or be a roadblock to getting drugs into clinical trials. Yeah.
Okay. So, and I appreciate
that. Well, I think that's what you just said.
I appreciate that Dr. Woodcock provided that quote. Yeah, I think that's spot on. Again, the balance between making sure that the drugs are safe because they're going into patients for the first time and having them available, getting them into the clinic.
I think that is one aspect of a general way of thinking of phase-specific development. So through all of the aspects of CMC, the requirements evolve as the product goes through different phases of development. The expectations become greater because you're learning your manufacturing process, you're learning your drug, and as you get closer to marketing, you need to have a robust handle on it. And there's a learning process. And so at each stage, the expectations become a bit greater. So I think as we talk about the different criteria for drug development, that will be a theme.
Yeah, I was thinking back when I was brand new to the FDA, my supervisor first week dropped a stack of guidance documents and the CFR and said, "We'll get you an assignment soon, start reading these." And then after a few days of reading them, I went back and said, "So can we talk about this?" And he said, "Let me boil it down to you. For a phase 1 IND, there's a medical need, otherwise they wouldn't be developing this product. Okay? Would you be comfortable giving this drug to your grandmother, to your kid, if they had this disease, if it potentially could help us learn about this disease?" That's a general goalpost to look at. So, the manufacturing process and controlling that and being able to document it is a very important part of that. Do you have confidence in it? And to your question of, can I make this on my lab bench, when I think about when I was a grad student or a postdoc, not on my lab bench. It was not controlled to that level. Right. I think more realistically, though, is that if you think about a drug development plan, you do discovery work, you get targets, and then you start to manufacture the drug substance. And I'm more of a small molecule reviewer. So a small molecule synthesis at a graduate lab or even a large research lab, you're making gram quantities. You're going to need enough drug substance to do your non-clinical studies and eventually to go into patients. Well, if you're manufacturing it at a lab bench scale, you're probably not manufacturing it at the scale that you're going to need to go through. And if you go batch to batch and start evaluating them and they have different impurity profiles, you're going to question whether the non-clinical data is really supportive of the proposed clinical batch. There are advantages of using some of these companies that are more versed in GMPs and more capable of scaling up to gear up to a clinical trial.
And I think we're going to get to that with the impurities, degradants, later your tox batch versus your first in human clinical batch, and considerations and caveats with regards to that.
But Jeff, I'm sorry for interrupting, but I did have an example come to mind as both Wendy and Olin were talking. And Wendy, you had mentioned the phase-specific requirements. As a microbiologist, I want to share one example in terms of expectations at phase 1 versus expectations at marketing. I mentioned the sterile drugs and the requirement for drugs that are going to be injected into our bodies to be sterile. We expect at phase 1 that the manufacturer is, of course, implementing sterilization processes, right? So that when we're assessing that IND, we're making sure that they're manufacturing that drug with a sterilization step. We're making sure that all of the components that interact with that drug and the container closure system that it goes into are sterile. However, what we don't require at phase 1 are validation studies and validation data to support those sterilization process steps. So that's a difference between the requirements at phase 1 versus the requirements at a commercial marketing application stage. When that NDA or ANDA or BLA hits our door, the application will not be approved without the necessary validation work to show that those sterilization processes, in fact, are adequate. So that's a big difference, isn't it? What the guidance points out is that if you are making a sterile drug, you should be doing all of those things, But the difference is in the IND submission itself, the sponsor is not required to supply a lot of data and associated analysis. So, you know, Wendy, when you went, sorry for repeating myself, but when you mentioned the phase specific, that's one thing that comes to my mind. And so, Jeff, your comment about helping to get drugs into the clinic, that's one example right there.
So John, just to clarify,
Yes.
the testing has to demonstrate that it's sterile.
Correct.
But the process doesn't have to be validated.
Exactly right. So we would still expect sterility test. If there is a requirement for an endotoxin specification, we would expect an endotoxin test as well. And we would expect, we would look for all of the sterilization steps. The sponsor needs to say, okay, this is how we're manufacturing the drug. We're going to filter sterilize it. We're going to autoclave it, whatever it may be. But the difference is all the details in terms of their validation data.
That may be a difficult concept for these early stage
Yeah.
investigators. The idea that, well, I have to have a test to show that it's sterile, but it doesn't. But what is this validation part? And the validation part is a whole big endeavor, right?
It's a big project. Yeah.
Which they may not understand the difference between what's the difference between testing it's sterile and validating that it's sterile.
Okay. Well, the concept of quality by design. If you think about quality by design and making a drug with good quality, with all of the manufacturing steps up front leading to the quality that's built into that drug. From the micro perspective, it would be demonstrating that the processes that you use to sterilize that drug all work and that they are adequate. And I'll back up just a bit and say that in terms of manufacturing a sterile drug, there's really two different approaches to that. There's one that's referred to as aseptic processing, where the drug solution is filter sterilized, and each of the container closure components are sterilized separately. And then in a very controlled environment, ISO 5 environment, which is pointed out in the guidance that we're talking about today, those two sterile components are brought together, the sterile solution into the sterile container enclosure. That's referred to as aseptic processing. There's also a separate process called terminal sterilization, where the drug solution is placed into the container closure, and then those units are either autoclaved, so a moist heat process is used, or gamma irradiation. There are multiple ways of terminally sterilizing. So my point, Jeff, is that whichever process the sponsor is using, the validation piece is showing that those sterilization steps work. That's the validation process.
A question along those lines. One, many
Yes.
times small molecule drug sterilization, aseptic versus
Yes.
what Wendy works on, and biologics, there are differences there.
Yes.
So we can't, our products
Terminally
cannot
sterilized.
be terminally sterilized. So it's a matter of
Understood.
controlling the raw materials, the process.
So a dumb question though,
Yeah.
as John just threw out gamma irradiation. What happens if I take a monoclonal antibody and gamma irradiate it to sterilize it? Does that cause enough changes to my protein that it's not functional anymore, or it has issues?
Well, I would say if you did it, you'd have to demonstrate that it didn't
And
impact
it
your
did not
protein.
do that. Okay.
But your point is well taken, Jeff. There are many drugs that cannot be terminally sterilized. I think that's the point that you're both getting to, right? Whether it's the heat denatures the drug itself or the irradiation process. So for some drugs, terminal sterilization is off the table, which drives them to the aseptic process, the filter sterilization and the separate
That's
sterilization.
more quality by design?
And again, the
Well-
raw materials, making sure that everything you're starting with is very well controlled.
Correct. And I would say, Jeff, that the quality by design pertains to either process. You're building quality into the process, whether it's a terminal process or an aseptic process.
So let me summarize for some of our
Sure.
investigators.
Please.
Do all the discovery work and early phase, not GLP, preclinical tox studies, but your early stage toxicology, your modeling, your animal studies, that are all whether or not you want to move a particular product forward. That can all be done with synthetic product that you've made in the lab, or that a biologic that you've made in the lab. But once you're going to take that into once you've decided this is the candidate I'm going to move forward with, the idea of making that drug product in your lab is kind of a non sequitur. At that point, you are now looking for a facility that can do it according to GMP guidelines. These phase 1 GMP guidelines. Would that be a reasonably fair summary?
It could be. I mean, I at least don't review the product until they're proposing for it to go into humans, right? So I think the bigger question would be, you should ask that of the non-clinical folks.
Yeah, I mean, all the discovery stuff.
GLP requirements?
But I will
What are
say
their
this,
requirements there?
that one of the very important things to look at to link the non-clinical studies with the clinical material is the quality attributes of the product.
Yeah, and when I say
And so
non-clinical,
if
I
you're-
don't mean non-clinical GLP IND enabling. I mean your early stage discovery.
okay.
Many
Yeah,
folks,
just to know
many,
if you
think-
want to carry
many
it
folks do
further.
tox studies that are just not GLP tox studies.
Okay.
They're doing or activity modeling studies that will not necessarily be IND enabling. Those are the studies.
So you're talking about characterizing a product to know whether you want to carry it further in
Exactly,
development.
whether it's a no-go based on,
Right.
on laboratory experiments. Yeah. Have you guys, just to throw this out, because it kind of fits in here, where we have this early phase, good manufacturing principles and increased regulatory flexibility, but for certain products, not necessarily for CDR products. But have you guys seen where you have that CMC activity disconnect, say for an ADC, where all of a sudden in the first human trial, because the ADC is so effective, that you're seeing, you're seeing 40% complete responses in a refractory patient population, where there's a disconnect because we're trying to facilitate, early entry into the clinic, safely, appropriately, with appropriate safety. But now the activity is so striking that the next 10 patients might see an accelerated approval BLA. I mean, have you seen that for a biologic, where there's striking early activity, yet the CMC development plan is now kind of disconnected from the substantive activity that you see early?
I think there's enough examples where the clinical development is outpacing the CMC development.
Yeah.
And I'm thinking of one example in particular, which is not the first in human studies. It was at an NDA stage, and there was a late signal that showed that this could be very useful for pediatric patients. That's very different than the first in human studies, but what ended up happening is the company had to develop a pediatric formulation. Very young patients couldn't swallow tablets, needed an oral solution. And we worked with them in real time to develop this, while we were reviewing the NDA for their tablet or capsule formulation. But in those cases, a lot of the concerns of impurities from the drug substance, they had been characterized, and you're not changing how you're making the drug substance or its scale. The question came that when you were changing the formulation and changing its storage condition and its container closure, was it still going to be stable? Not just the chemical falling apart, but was it going to stay in solution? So we've seen such situations like that. I'd say that's one of the roles of the reviewers once it has gotten in past the first in human, is as the clinical development goes forward in scaling up the manufacturing process, moving towards a formulation that they're anticipating to be the commercial formulation, that we identify showstoppers before the NDA even hits the door. But usually it's at that point, it's not at the first in humans stage.
But I would say that we do deal with that in the context of breakthrough products. where it can be an accelerated program. And the manufacturing process is something that evolves. For the I'll speak only about the biotech products, but the manufacturing process is always evolving, and the knowledge of the drug is evolving, and it takes time, and sometimes you have an accelerated process where it doesn't percolate through the same way, and that's a scenario where you really want to be meeting with the agency on a regular basis, and that is a feature, right?
Yeah.
Where you can meet with the agency on a regular basis to make sure that you're on track and that you have what you need.
I wish we saw
Yeah.
it more often, but it definitely happens where there's substantive early activity. When, hence the reason for the breakthrough therapy designation in the first place. And it seems like there might be some CMC activity disconnect and that's why those programs are there and why you guys provide additional help to try to get the CMC up to par.
So it's always good early in your development program, even though the expectations are different at phase one. It's always good in your development program to think about the next step so that you can be anticipating what you're going to be needing.
And I don't know if biologics participates in the CMC readiness pilot, but that's one of the programs that OPQ administers to make sure that if you have a quickly developing clinical program that we can head off any CMC manufacturing process.
What's this called and how do people access that?
So I don't get to participate as often. It's called the CMC readiness pilot. I believe you'd have to Google it, but there are submission pathways where we take a deeper look at what you have in-house at the time on their application and try to project in the future, if as they scale up, are there going to be issues that we want to head off before the NDA hits the door? All of which is to make sure that the CMC information is not the rate limiting step.
And presumably through breakthrough therapy designation, that same kind of scrutiny and effort is being...
You get a lot of the same benefits through breakthrough designation, fast track, the emerging technology team, the CMC readiness pilot. They all have additional interactions where we take a deeper, closer look at what's there.
Great. So this may be a place where I know we want to get to critical quality attributes, because that is a question that comes up all the time from early stage companies and investigators is, what do I have to do? But it sounds like this might be a place where maybe the tox batch and the clinical batch, what the advantages and disadvantages are of for my GLP toxicology studies, when I found my CMC CRC manufacturer who's going to make my drug for me and presumably maybe make the tox batch, and Olin and Wendy can talk about this for small molecules, and John as well. I have told folks in the past that it's best to take your GLP tox batch and make enough of that, that the tox data that you get and the impurities that are in there are qualified so that you can use that in your first in human trial. But there are advantages and disadvantages to that. Maybe you guys can highlight those. Why maybe sometimes the more, I think Olin's explanations in the past have been, well, the more batches you make, the more experience you get with making those batches, and therefore the better those batches ought to be, not, so I'm going to let you guys run with that for a bit.
For small molecule products, it's not uncommon to see an IND hit the floor with one specific route of synthesis, okay? And depending on how developed it is and understood that process is, you'll get an impurity profile, 10 impurities at varying levels. And I think what you're referring to is a concept that's described in the phase 0, phase 1 guidance for product development, where they say if you take that and use it in a tox batch, presumably the profile, the 10 different impurities, are going to be the same impurities that would go into a clinical batch, in theory. The problem is you're going to have to manufacture that drug substance batch. It's going to have to be stored and wait while you're doing the non-clinical studies, and in that time it might degrade. You might get an 11th, 12th, 13th impurity, or some of those 10 impurities might grow over time. You may not know the best way to store it. It may have needed to be colder or whatnot. So your risk there is that by the time you've done the animal studies and found what levels are qualified for each of those impurities, and then take that same drug substance batch that may have degraded and then analyze it, well, it may have degraded on its own, or the processing of it in the drug, into the drug product as you formulate it with excipients may cause other impurities to arise. And now you have new impurities or higher impurities that have not been qualified. So, on the other hand, you can manufacture your batch for animal studies and at the same time develop or improve that manufacturing process. So you have discovery chemists, medicinal chemists, and you have these wizards called process chemists that get much better at looking at something. When we want to go from a gram to kilogram and multiple kilogram scales, there are simpler, better ways of making it. They're real wizards and good at their jobs. And the advantage is that generally we see better synthetic routes with fewer impurities or lower impurities. In this case, your risk is you have a new impurity from those original 10, and now you have to do that animal study again to qualify. So that's a business decision. We don't make business decisions for the applicants or for the investigators, but those are your risks, and you can do what's best, whether it's to invest early on in improving the synthetic process and the drug product formulation so you understand it better. Much less likelihood that you have to go back and repeat some steps, or you try to go a little bit cheaper and cross your fingers that you're not going to find new impurities as you move along. Yeah. Those become the hold issues later in IND review. Similar experience?
And I would say those concepts apply also, those principles apply also to biotech products. I think that if you wait until you have, in addition, if you wait until you have your GMP process, it takes a bit longer. So you can start getting your information before you have your clinical batch, and you can leverage the information from the toxicology batch in terms of stability, something that Olin was talking about. When people come in for phase 1 studies with a GMP batch or limited numbers of GMP batches, there's often limited stability data. But if the product is sufficiently comparable, you can leverage information, your stability data from your tox studies that helps support your first-in-human study and the use of the clinical material.
So there's a couple thoughts that popped into my head as you guys were talking, and I want to just throw those out there. If I can remember them, or they're going to be gone. But one of the questions was, I have seen this a number of times, is, well, how many batches do I need to make before I can go into the clinic? And what would be the kind of informed, intelligent, thoughtful drug development approach with regards to the number of batches that would probably be wise to have produced before you were to go into clinical use?
You want me to start with biotech?
Yeah, go ahead.
Okay. So typically we would see toxicology batches and then limited GMP batches, right? If you have the more batches you've manufactured, the more you understand your manufacturing process, the more data you have for setting specifications. But we don't want to hold up product development. So if you have a single batch and you have enough for your clinical trial, you can do a clinical trial. You may not be able to proceed with your next trial until you have more material, in which case you would have to establish comparability. If you have only one batch and that's not enough to complete your trial, that's a risk, because you may get into your trial, not have enough material to complete it, and you don't know for sure that you can make another batch that's comparable. And so because there are impurities, there can be product-related impurities from the manufacturing process, also from degradation. You need to make sure that your product is sufficiently comparable, or you won't be able to pool the data if you have different product that goes to patients in the same study.
So this is probably a place to also talk about bridging, because this sounds like a bridging concept here as well. Bridging the product, one product to another product, changes in that product.
So the idea is that it would be the same product, just different lots of the same product, but they have to. So this introduces the concept for the biotech products of heterogeneity. and I think you may be talking about this as
Yeah.
well.
I think it's a bigger issue for you than it is for me.
Right. So the biotech products are manufactured in living cells, and largely have depending on the system you're using, they'll have post-translational modifications. And so what you'll end up with in the end is a heterogeneous mix. But you want defined heterogeneity. So you might have different post-translational modifications within that volume of product. But what you need to assure is that from one lot, first of all, that it's stable and that you're not changing the levels of those variants. But then also that you can make multiple lots and have the same ratio of the same component mix, that it's controlled heterogeneity.
You're retaining samples for all of these different manufacturing batches.
And that's a different concept. We're going to another, I mean, it's the same, it's based on that, but that's a reminder to the sponsors to hold retained samples so that you can bridge manufacturing processes, you can bridge assays.
So I hope to get to critical quality attributes and what are the important ones and which ones aren't and what you have to measure in a second here. Owen is looking at me.
But for different batches, the idea is if you go to multiple lots, you need to ensure that they are consistent, because otherwise you can't mix them in a clinical trial.
Yeah. I mean, we do see this some in the small molecule world, not with the simple solid oral dosage forms as much, but for a more complicated product like a liposome or a lipid nanoparticle, the manufacturing process is very important in the final drug product. And this is where investigators really have to balance what are their objectives. You know, if the first in human study is just a demonstration that it is likely a candidate that deserves more investment, you might approach this differently and just try to demonstrate this and then sell off your product to someone else that's willing to invest heavily into understanding the manufacturing process. But if you have a product that has multiple components, it has a lipid shell, it has a targeting moiety, and you're loading it with a drug substance on the inside, there are a lot of parameters you have to control to get consistent loading within that liposome, consistent density of the targeting ligand on the outside. And so to Wendy's point, a lot of development of that process and the analytical methods that go into monitoring it are evolving while the clinical study is going on. So you can see examples where later in development we've changed the sites, we've changed the equipment. We're trying to demonstrate that we're measuring 7 different quality attributes and show that it was the same as our early studies so that you don't have to repeat safety studies. That would be a really expensive proposition. So saving enough samples and storing them appropriately so that they can be retested is important. And similarly, if you change the methodology enough, you may have to go back and demonstrate that bridge from your new process and your new analytical methods to those earlier batches and those earlier methods.
I watched a presentation recently where there was a sterilization technique where they had filters in series and they changed, or they were in parallel, and then they changed those to in series. You know, and this was for an ultra rare, disease, and it was a biologic product, and they didn't think that change was a big manufacturing change, and it wasn't necessarily pointed out to the agency, and it completely, literally put their whole product on hold because of sterility issues and product quality issues. So let's move on to from manufacturing in batches and retaining samples to a question that come up a lot is, and I've listened to these guys, opine enough that I can even say the words, but how do I know of all these critical quality attributes of my, what did you call that, chaos, heterogeneity, how do I know which of these components, which of these attributes I need to measure for my my potency assay, for my product quality? I mean, those are kind of complicated questions, and Olin is going to say, know your product. And I don't want to steal this thunder. I'm going to let these guys talk about it. But I think it's really important is that you're the one who's developing your product. You're the one who made certain changes to FC receptor binding, for whatever reason, to accomplish certain, an end game here. And those are probably reasons. Those are probably components of your critical quality attributes. Why don't you guys run with that?
I'll start with saying that John mentioned quality by design earlier. And there's a ICH guidance document Q8, and it goes through a couple of concepts. But one way of framing it is working backwards and starting with a concept called the quality target product profile. And this is, if I had my ideal drug, this is what it would look like, this is what it would do, this is how stable it would be, this is how what the dosage form would look like, and what its strength would be. And one way to demonstrate in the IND submission that we can trust you is to demonstrate that we've really thought through what the end goal will be, whether it's for that first formulation that you're just going to try in humans for the first time or at the marketing stage. And once you have that target product profile, you start to say, well, what can I actually measure to demonstrate that it meets that product profile? And those are your critical quality attributes. So if you read the regulations, it says that you have to demonstrate the identity, the purity, the potency or assay is how we use it, and its quality. And quality is an intentionally nebulous term because legislators don't necessarily know what creative products are going to come out. And so as I mentioned earlier, one of the enjoyable aspects of my job is sometimes I get nanomaterial products and I get to review them, and they truly are novel. And at that stage, we may not know what is going to be important to control years down the road when you're marketing. And so how are you possibly going to bridge back to it? And we've had this discussion, Jeff, where I say, in many ways, there's much more characterization and testing for those early lots. When people think of nanotechnology, they think size, and in the early days of the nanotechnology products that we saw, and people were measuring size in one way. And it turns out that sometimes size is a surrogate of what's actually important. Sometimes it's the amount of surface area or the porosity of a compound or of a material that's more important. Some of those better products that we've seen come in the door actually came from material science labs where they were trying to find a use for their product. They characterized the wazoo out of it. At the time when they come in for an IND to put in humans, they may not know what an appropriate range is appropriate for its size or surface area, porosity, its zeta potential, or measuring its charge. So at a minimum, we ask them to report what it is. And so if we can go back in time and look from batch to batch. If we have a general idea of what has been manufactured through multiple batches, generally we ask them to put ranges on those different quality attributes, both in terms to demonstrate that it is consistent, but also as a check of if something falls outside that range, that tells us something went wrong with the manufacturing process. And that's just a red light for us and the investigator to dig in and say, what happened here? Is it likely to have an impact on the patient? So I'll pause and see if you two have anything to add to.
You have said this before, but early stage, you may be actually looking at more critical quality attributes than you are at later stages, because the early stages you're trying to identify those that are actually truly important or more correlated to activity or potency or purity, but those aren't all validated assays, right? Those can be reasonably less rigorous and burdensome assays that you're conducting with those multiple attributes that you're looking at. Is that a fair statement?
Yeah, like John was saying with the validation of the sterility process. The analytical methods are under development, and we know that. They are not ideal. As the drug substance manufacturing process changes, it may need to be the analytical methods may need to be modified to detect different impurities that weren't present in previous batches. So we understand this. There are minimal layers that are expected to demonstrate that it's specific for the analyte you're looking for, that it's linear within the ranges that you're evaluating, that there isn't interference between impurity peaks and your assay peaks, and that it's reasonably rugged to variations in whether you're the one, the analyst doing the analysis, or if I am, if we move different labs, or if we do it under different temperatures and such.
So one more question. We're going to get to potency, but before we move over to potency and critical quality attributes for biologics, I just want to kind of continue with small molecule drugs, and that your potency is like strength, kind of.
Assay is usually the term we see, right? Because small molecules are not, unless they're a peptide or an RNA that's likely to take on a secondary or tertiary structure. Small molecules, if you can analyze it on an HPLC, it's not going to have a fold that's going to impact its impact in the body or its potency.
But along those lines, this is where drug development is a complicated endeavor and an enterprise. But small molecule drugs always seem reasonably, easier or more straightforward. But when it's no longer milligrams or micrograms of a small molecule drug, but now it's a nanoparticle with a targeting moiety on it, with different components in the nanoparticle, and there's multiple components potentially inside this nanoparticle. At that point, you just can't measure a milligram quantity of nanoparticle. I mean, the characterization of that product is complicated.
Sure, it gets more complicated. I think that's why we see it more in the oncology products. If you're going to go through the hassle of making something that's more complicated and more challenging to characterize, you better have a good reason for it. But even so, we have examples of what we would expect to see in something like that. So we can back up again to the quality target product profile. I want a particle that has a certain amount of drug within it, whether it's a liposome or a lipid nanoparticle. I want to have targeting moieties. How dense does it need to be? We define that. How much of the drug is encapsulated and how much is on the surface? And there are ways to separate those, and it's in the literature where you can get a starting point for that analysis. And then other things, its charge and its lipid compositions. Some of those are starting to become more standardized.
When I think about potency assays for biologics, I think of mechanism of action and activity. But do we ever expect, like if you have an siRNA and a nanoparticle, that part of that strength or assay is that does it downregulate, does it affect the mRNA in cells? Or if you have a protein degrader, does it degrade the protein in cells? Or is it more the constituents, how much of that protein degrader is in the nanoparticle, how much siRNA is in the nanoparticle? But the actual, what it does isn't part of the, for biologics, when we're going to get there, it's actually what does it do? Is it doing it, and how well does it do it?
Right. So this might be a good time to jump over to bioassay. Mm-hmm. Do you want to go from there? I would say that you bring up the example of siRNAs. In this case, the mechanism of action is known, and it's known to the point that if you control the assay and just the content, you don't need to bridge that to its activity. Because if we know that it is there, if it's duplex RNA or single-stranded RNA, and it's stable, then the assumption is that its activity is not going to be impacted by other aspects like its folds. Mm-hmm. I think if you got longer than its typical siRNA, then you might start to see something that requires some sort of bioassay.
Yeah.
Go ahead.
Wendy, critical quality attributes and potency.
Okay, so just to go back to testing that you asked, what do you have to do for the start? So safe, pure, potent product. That's the goal, right? So there's always safety testing that has to happen. And I don't know if you want to get into this now or go back to manufacturing later, but you have to demonstrate that you're starting with non-contaminated material. You've got to do all the safety testing. You're not introducing viruses, you're not introducing mycoplasma, you're not introducing any infectious agents. The contaminant levels are controlled. For purity, that's where we get into critical quality attributes. And so again, it's heterogeneous. And this is the kind of thing where your knowledge is expanding as you go through the the process of the IND process, the development program. So when you design your test, so as you said, you start with a product, because Olin mentioned this, you start with a product, you have an idea of what your product's going to be and how you want it to work, what you're aiming for. Do you want to activate a pathway? Do you want to block a pathway? And these are protein products, and they have, you know, multiple layers of structure that can impact their activity. They may have multiple mechanisms of activity, and all of these are going to be detected on some level by physicochemical characterization, but critical to this, and also part of the regulations for biotech products and biologics in general, is the need to have a potency assay. That is how our products measure, a bioassay. That's how our products measure potency. It's not the level of material. It's not the amount of material. It's the activity, and it's a readout. It's a readout of activity. Generally, we like to see bioactivity and cell-based assays. But again, the assay, you design it on what you know about the intended mechanism of action, the expected mechanism of action. So that and the assays, I just want to make a comment about the need. They're not validated until the time of licensing applications, but they have to be qualified and they have to be demonstrated that they are suitable for the intended use. And that's, I think, what you were getting at when you were describing what you needed to understand about the assay. So in terms of what endpoints you need to look at to identify critical quality attributes, you run physicochemical variety of tests, and you look for charge variance and size variance and contaminants, purity, aggregates, fragments, and you try to understand how each of those different attributes, different parameters might impact your mechanism, your product's potency. And so that's, and you're going to be learning as you go along. So you might start with an assay that shows you a range of charge variance, and you're going to want over time to understand what the makeup is of each of those peaks that you see on your chromatogram. And it may be that some of them lose activity, it may be some are equally potent to the product that you've defined.
Would I have to do this before I got into my first in-human trial, or this is during?
So you're
during
learning
development?
this. You're learning this, and you start with what you expect. You start with what you expect to have, and you're going to learn this also from stability studies. You might see some breakdown of samples that have been stored, of your tox data, of your and then you try to identify what's the impact of that breakdown. What's the impact of that other peak that's coming up. And so over time, you'll eventually want to purify it and test it for potency. And so you learn as you go along. You start with everything you have. And as long as you're controlling it and you're getting similar numbers while you're early on in development, certain consistency, then you know that you can get information from your clinical trial. But as you go through development, you want to understand each of those peaks better, so you know what really needs to be
So
controlled.
this is the physiochemical characteristics of the drug product.
Yes,
that
and
may
you
be
relate
changing
them
over
to
time,
potency.
and you're trying to, you'll see those and try to correlate those back to activity or.
Yes,
okay.
you will correlate those back to potency, and that will tell you if a peak is, maybe you're getting what looks like a contaminant, an increased peak. You find out it's oxidized. Maybe it's it could be anything. And then you look to see, well, maybe you lose activity, maybe you don't. If you lose activity, you're going to want to control that peak. And so for release, you're not going to want as much of that peak. Stability, you can't let that peak get beyond a certain point, or you'll lose the potency of your product. So you're learning that as you go along.
Now talk about the actual potency assay and what the purpose of the potency assay is, what it's really supposed to measure, and then earlier on what are acceptable alternative potentially substitutes or potency assays that may not necessarily completely define the mechanism. Of action.
Sure. So you want to go for a potency assay that establishes your presumed mechanism of action of the product, and to the extent that you understand it. And there may be more than one mechanism. And so in that, you may need more than one potency assay. So these are pleiotropic molecules. They have multiple ends that can be doing, different business portions that can be doing different things. So you would want to characterize, so in the straightforward case of an antibody, you have the FAB arms, you have the FC fragment. Sometimes the sponsor will engineer out FC binding. You don't need an FC effector assay for that. But it may be it may be the sole role of, the key function, the key mechanism of the antibody that might be, so you have binding and then you have the cell death. So you have to understand that. Now, you may be able to start, the ideal assay, these products, and I'm starting with antibodies as just a straightforward example, but we have a lot of different kinds of biotech products. So
which is so
you have
cool
to
that we have
adapt
ADCs
it, right?
and multifunctional
and
engagers and
cytokines and growth factors and right.
Was the idea of what the mechanism of action of the versus that sometimes binding activity might be.
So a good potency assay, so if you have something, it needs to engage, right? It has to engage your target. So a bioassay would have to, if you're looking for an endpoint that's, say, maybe cell death, it's going to require that the product bind the target before anything else can happen downstream of that. It may be that you can start with a binding assay, but you want to have a plan. Ideally you would have a bioassay that captures the endpoint that can be a reflection of the mechanism of efficacy of the product. So if you start with a binding assay, it has to be a relevant binding assay. It has to bind your target. We have seen products come in where they're binding something, but they're not binding the target. That's not going to be, that's a whole issue right there. That's not going to help you capture the potency of your product. So you design it, and it may be that the assay evolves over time, but it is highly recommended to get as close as you can to the kind of potency assay that you want early on. Because again, if you don't, there's going to be bridging on down the road. And so if you're changing your potency assay, you're going to have to, that's a really clear case where you need retained samples. You're going to have to be bridging it to the earlier samples and show that your assay is capturing the same information or more.
So it seems like, potency assays, particularly in some of across centers, can be extremely challenging. But, for CDER and for, biologics, many times I'm assuming that these are cell-based. That, okay. Preferably you would try to develop an assay, and I have seen some over the last 23 years, where they were actually animal-based potency assays, which seem very complicated.
Those are very difficult.
Yeah.
Right. So you want to be developing an assay that can be scaled up, that can be, can be high throughput. If you need high throughput, that is adaptable to that. Animal assays don't necessarily fit that. Now there may be scenarios where you need an animal assay, but it depends on what your product is. But for the most part, those are not. Those are very challenging. So cell-based assays, we tend to see more. What we usually see are cell-based assays, and those should be able to capture hopefully we'll be able to capture the mechanism of action. You may need to use a mixture of cells. It may not be a single cell. You may need interactions of cells. A lot of our products form links between different cell types or between a target and a cell type, and so there may be.
That's it for our multifunctional engagers.
Sure.
The potency assay could be potentially complicated and not always easy to.
But much more straightforward than animal assays, and much easier to control.
Okay.
Yeah.
There's so much to talk about. I'm not sure, and I don't want to be completely tangential, but one of the, since I was thinking here, gee, I have an assay, do I have to have a master cell bank for my cell line that I'm using in my assay so that my assay stays the same? But as I thought that, I was thinking about, manufacturing, and when I'm developing cell lines and my master cell bank, to have my, my clone that I use to manufacture my product, my ADC or my engager. But there was a new concept that I came across, which I didn't even know about because I've kind of not been in the trenches of the review process. But this idea of, instead of having to have a master cell bank and a clone that I'm using, that I can use a polyclonal, multiple cell lines to make my product for first in human trials early. Have you seen that?
So you mean starting with a cell line that has not been cloned?
Yes, exactly.
That
I can have a pool of cell lines, and my product comes from that cell line.
So I want to just distinguish between pooling cell lines versus a parent cell line that hasn't been cloned.
I'm not sure. Educate me.
So I mean, in one scenario, you might have, I think we're talking about, in one scenario you might have, you want to make a mix of products, and you might have different cell lines that you then mix the product. And so you're ending up with polyclonal, you're ending up with a mix of antibodies. Again, we're using antibodies as an example, as a model here. Versus, I think what you're asking about, which is a cell line that has had your gene of interest introduced, and usually we like to see a clonal cell line. We like to have that cloned so that the product that you're using in your clinical trials comes out of a clonal line, right? Because then it's more likely to be consistent in terms of the attributes. And I think what you're asking about, but that takes time. Right? And it can slow down development.
Right.
And if the product's not going to work, it's a lot of time that you've gone into it. So I think that's what you're asking.
Exactly. Yes.
Can you take a cell line that is producing your product, but it's not clonal,
yes,
and can you use it?
yes.
And so I would say, so the answer is yes, we have seen it. Can it be done? It can be done for early studies with the expectation that you will need to address how you're going to characterize the product that comes out of it, because eventually you're going to need to clone it. So you're going to, again, need to bridge the product that comes out of this polyclonal line with what comes out of your ultimate, what you ultimately derive as your clonal line. And so that's bridging. It's also a similar context to what happened. We've always been concerned with proper cloning. I mean, so I'm going to take this a step not quite as far as not cloned at all, but if you were to take a cell line that maybe isn't properly cloned, and there are some contaminant subvariants in there, subclones in there, over time, and over time, you can imagine that those subvariants, those clonal variants might grow out, and that could change your product, and we have seen that. And that's the kind of thing you don't want to have happen late in development. That's a real problem. And so it's a similar kind of question, because you could be getting a lot of different variants that you can't control the same way, because some might grow differently than others.
There's a risk involved.
There's a risk involved. But if you were to do it, you would need to have a plan for how you're going to bridge that information that you've come out with from your early studies, your early trial results, with later studies with the clonal, with what comes out of the clonal line. So there's a risk, and it kind of puts a greater burden on you later.
This critical quality attributes concept is critical, and I know that you have referenced a number of guidance documents that really help provide some thoughts around what to think about with regards to critical quality attributes for biologics. But one of them was the points to consider for monoclonal antibodies.
The points to consider for the manufacturing testing of monoclonal antibodies, it's, I think it came out in '97. It was really a very forward-thinking document, and it really lays out, it's focused on monoclonal antibodies, but the principles really apply to biotech products in general. And it really helps it doesn't cover everything, but it does provide insight into the kinds of questions, the kinds of things that the reviewers look at, and the thinking behind it, why it's expected. I highly recommend that. Yeah.
Does the OPQ for small molecule drugs have kind of general guidance with regards to consideration for critical quality attributes that might help provide some concepts around what to think about, that we've already just discussed here?
Sure. I mean, like the older guidances for the phase 1 and phase 2 and 3 development guidance for INDs talks about, they don't call them critical quality attributes because it predated ICH-Q8, but they give examples of different attributes that need to be measured and monitored. Q8 takes you through that process of thinking through of what might be important. I would point more to some other guidances like liposomes are a specialty sort of formulation, and there's a special guidance for liposomes that that was written and revised, I think, within the last 5 years. The products that contain nanomaterials is kind of a thought process sort of document. It takes varying chemical and physical properties and describes what kinds of analyses might be necessary, what kinds of physical or chemical attributes that need to be monitored.
So, as you mentioned nanoparticles again, and thinking about attributes to look at, but this is a point where you should probably highlight the Nano Day symposiums that you guys have produced.
Yeah, there are a couple of presentations that are available on YouTube. I think the first one we did with OCE that you helped coordinate for product development, and I did that with my colleague Parashmik Patel, who went more in depth into the drug substance development. I went into the drug product development, and Wendy had a segment for biologics. But there's also the FDA Nanotechnology Task Force, and we bring all the centers across FDA to discuss various activities, whether they're review or laboratory work that's being done in the nanomaterial field. And we also discuss funding mechanisms that go into some of those basic research projects. One thing that we do is every year on Nano Day in October is we have celebrations. This year we're doing an internal celebration. It's going to be more of a poster session so that we can talk about proprietary information that we can't share. But in previous years we've had external speakers come in and talk on projects such as continuous manufacturing of lipid formulations and lipid nanoparticles. And those are available online.
Jeff, if I may, I'd like to speak a little bit about the microbiological critical quality attributes, in addition to the CQAs that Wendy and Olin just described. So from the micro perspective, for sterile drugs, at phase 1, our expectation is that the drug is sterile, and we had talked a little bit about testing earlier in our conversation. So I'd like to just refer our viewers to USP 71 for sterility testing requirements. If the drug is intended for injection, in addition to sterility, it needs to have an appropriate bacterial endotoxins specification. And I would point our viewers to USP 85 in terms of how to best go about setting an endotoxins limit. For ophthalmic drugs that are applied topically, there's no endotoxins requirement. However, they need to be sterile. For an ophthalmic drug that is injected, it does have an endotoxins requirement as well as a sterility requirement. So those are CQAs that pertain to microbiological quality for the sterile drugs. For the non-sterile drugs, I would point to our recently published 2021 draft guidance from CDER on microbiological quality considerations for non-sterile drugs. And after our conversation, I will send you that,
Okay.
that guidance so that you can share that as well. But
The ophthalmologic one, I'm not sure that we need, only because I haven't seen, in all my 23 years, I've yet to see an ophthalmologic drug that's been used for oncology.
and that's, from my perspective,
no,
I
absolutely.
deal with all of the
Absolutely.
drugs that come into
Yeah.
Cedar, so
Yeah.
I'm just going through my list in my head. But I'm glad you pointed that out.
Guys, this might be a place where we can quickly talk about major issues that you've seen that arise from CMC kind of quality product issues that put folks on hold.
Common issues are impurities that are not qualified. Either they are different from what the clinical batch is different from the non-clinical batch and you have a different impurity profile, or it wasn't qualified to a high enough limit in the non-clinical batch for what's being proposed in the clinical study. Okay, so there's one. Stability is another one, again related to impurities. I had one that is very light sensitive and it degraded over time when it was being stored, but it also degraded when it was being prepared for administration. So we have something called an in-use stability studies. So if you had a product that was stored in an amber bottle and in the dark, but then you prepare it for administration, and you find that if it's in a translucent IV bag and it degrades, well, you need to take steps to address that, whether keeping it dark or whatnot. Similarly, there are compatibility issues with once a drug is prepared for administration, it might adsorb to the plastic tubing and you're not delivering the dose that you think you're delivering. That's another issue.
And that, I'm sure Wendy will mention that because that happens with some of these bispecific engagers that the dose is so low that what do you call it, in-use
Compatibility.
in-use compatibility assays, that it just binds to the tubing and you don't get the dose.
Yeah, those are probably the main issues that we see.
Okay, so for the biotech products, safety testing, ensuring the safety of the cell bank, the testing the bulk drug, the bioreactor harvest and the drug, viral clearance, addressing viral clearance, so that and any endogenous viruses that are present in the cell line, because again you're starting with living cells, it can be adequately cleared. Sufficient characterization, showing if it's not sufficiently characterized, so you wouldn't know if you're doing something twice the same way. Linking the clinical material to the tox study, to the tox material, so that you can leverage, so that you understand that you're using the same product, that it's suitably consistent. Specifications, you have to have specifications. They can be broad early on, but they have to be meaningful. And this is where multiple batches can help. If you have a single batch, you're not going to limit your specification. We wouldn't expect you to limit it to just the batch that you have. But it could be broader than that. If you have multiple batches, you use your manufacturing experience to set your specifications. But they have to be reasonable. You can have leeway, but it can't be 0 to 100. You have to have meaningful specifications. In-use compatibility, as you mentioned before, and for the potency, it has to be a suitable potency assay. Also, stability data. If there's insufficient data to show that your product is stable for the time that you're using it, that can be an issue, a concern.
So I can't tell you how appreciative Project Catalyst is for you guys having taken the time to be able to do this. And usually this is the point where I would ask you, what advice would you give to these academic investigators in small companies? I'm going to summarize what I think you guys would say to these folks, and to make this quicker, but these guys spend a lot of time generating these guidance documents, and I would highly recommend that you step up to the plate and that you read the ICH and the FDA guidance documents, because they are extremely useful tools to help you understand how to think and what the appropriate questions are. The other components are to, one, interact with the FDA at every opportunity that you get. And there are more opportunities now than there have ever been. There is Project Catalyst, there is the OREG program to ask questions. These are the folks that I field questions out to, the subject matter experts, when you ask me CMC questions. Then there are also now, we have AID meetings, the Accelerator Innovator Discussion meetings, but there are Interact meetings that you can have. You can have Type D meetings. We have the pre-IND meeting, which, as Olin points out, is the freest lunch that you're ever going to get from the FDA. There are no PDUFA fees associated with that. And then at that point, if you actually do all of that and listen to us, you're likely to have a successful IND submission. So that's what I think these folks here would encourage the audience out there to do. And once again, I just want to thank you guys so much for taking the time to be here and have this great discussion.